Good morning, children. Today I'll take an experiment to demonstrate the process of plasmolysis and deplasmolysis in plant cells. In setup A, we'll take a fresh section of the leaf of an aquatic plant, Hydrilla, on a glass slide in water, and we will observe its cell under the microscope. Then in setup B, we'll take sugar solution, 10% sugar solution in a petri dish and transfer the above section of setup A of hydrilla leaf in it. We will remove this section after 5 to 10 minutes. We will mount it on a glass slide in the same solution and we'll observe various changes in the cell under the low as well as high microscope. This is setup A and this is setup B. Now in the setup C, we will quickly transfer the above section of hydrilla leaf before its cells die in hypertonic solution. We'll take it from that solution and we'll put it in another dish containing water, that is hypotonic solution. After some time, we will remove this section from the petri dish and we'll put it on a glass slide in water only and again examine under the microscope. So the observation is, when a fresh section of hydrilla leaf is seen under the microscope, the cytoplasm of the cell the cytoplasm of the cell is seen pressing against the cell wall. And there is no distinction between the cell wall and cell membrane. They are tightly packed. A clear large vacuole is seen in the center of each cell. The cell is in the turgid state. Now, when the section of hydrilla leaf is placed in hypertonic solution, as in setup B, the cell membrane along with the cytoplasm of the cell shrinks and the cell membrane will withdraw from the cell wall. The central vacuole disappears and a space is seen between the cell wall and the cell membrane, which is filled with hypertonic solution. The cells get plasmolized. This is a plasmolized cell. The protoplasmic mass, that is the cell membrane with cytoplasm, shrinks when this cell was placed in hypertonic solution due to exosmosis. So the protoplasmic mass shrinks and get concentrated somewhere in the center or side of the cell, leaving the cell wall. Then the space is formed between the cell wall and this concentrated protoplasmic mass. This space is being filled with the hypertonic solution. Then on returning the solution of hydrilla leaf to water or hypotonic solution, as in step C, the cells again regain their structure and the shape is observed. Will regain their structure and shape is observed as in step A. The same shape and structure the cell will regain as it was in setup A. The cell vacuole reappears in every cell. Cytoplasm is pressing against the cell wall. The cell wall is deplasmolized. We will not say it is turgid. It is deplasmolized because it is the reverse of plasmolysis. The re-entry of water has taken place and making the cell deplasmolized. This was about the experiment to demonstrate the process of plasmolysis and deplasmolysis. Now we'll do the significance of plasmolysis. Plasmolysis has great importance, use in fact food industries and some other field also. For example, when we prepare jams, jellies and some sweet soft drinks, we add sugar into it. Adding of excess sugar into it make the solution hypertonic, and we can preserve them for a long period of time without getting spoiled, because if the bacteria or the fungi happens to grow on these foodstuff, their cells get plasmolized because they have entered into the hypertonic medium, and the water from their body cells will move out into the outer medium. Their body cells get plasmolized and they are killed, they die. So first point, high concentration of sugar in jams, jellies, drinks, etc. protects them from decay. How and why? By bacteria and fungi. It is due to plasmolysis. The hypertonic nature of such food plasmolizes the cells of bacteria and fungi. Similarly, we can preserve pickles for a long period of time without getting them spoiled. Again, the same reason is there. For making pickles, we add salt and other spices in high quantity. So the high concentration of salt and other spices make the medium hypertonic. And if again the microorganisms like bacteria and fungi happen to grow on them to decay the food, it is not possible because of plasmolysis. Once these organisms get into it, they are in the hypertonic medium. So the water from the body cells of bacteria and fungi will move out from their body cells due to exosmosis. Their body cells become flaccid and the cells become plasmolized. The bacteria and fungi are killed. Similarly, meat, fish, vegetables, they are first dried and then salt is put on them to protect them against spoilage by bacteria and fungi. Again, when they are dried, we have removed water from these food stuff, and salt is applied on it. We have made the medium hypertonic. So if bacteria and fungi happen to grow on them to decay them, it's not possible. Again, due to plasmolysis, the bacteria and fungi cells get plasmolized due to exosmosis. Weeds, that is the unwanted wild plants, when they grow in the fields, they can be killed in a playground by adding excess salts in their roots. When we add excess salts in their roots, the water surrounding the roots become hypertonic. So the water from the root hair cells will move out. The water surrounding the cells become hypertonic. So as a result, the water from the root hair cells will move out. The root hair cells get flaccid and they are killed. So the plant dies and very easily they are removed from the playground. Plant growing in the walls, foundations of buildings are killed by adding large quantity of salt in their roots. It's the same case as we have done in weeds, removing the weeds. The root cells of such plants are killed due to plasmolysis. Excessive use of fertilizer in the fields can also kill the crop plants in the same way. Now, the agriculturists should know the amount or the quantity of fertilizer to be added in the fields. Excess of addition of fertilizer will not help them in growing the plant or in producing more of plants. Instead, the crops will be destroyed. It is because excess fertilizer outside will act as hypertonic medium. So the water from the root hair cells of the crops will move out due to exosmosis. the cells become flaccid or plasmolized due to plasmolysis, and their cells are killed, the crops die. Plasmolysis can be used to remove blood-sucking leech. It is a parasitic animal from the victim by sprinkling salt on the body of leech. When the salt is put on the leech, the water from the body cells of leech will come out due to plasmolysis, and their body cells get plasmolized. The cells are killed and the leech will detach itself from the body of the host or the victim, get relieved. Thank you, children. Tomorrow we will be doing the other part of this chapter.